Okay. So, good day, everybody. So, we're happy to have you here, and also particularly happy to have Professor Serdal here. Professor Serdal got a PhD in 2007 from the University of Texas, Austin, working with Professor Dmitry Makarov. Then he joined Ron Elba's group at Cornell University. And then he moved to Los Alamos National Lab as an associate research scientist. And then later on, he joined NYU Abu Dhabi, where he currently is. So he works on quite a number of things, but one of the things his group will be talking to us today about is about DNA condensation. So the topic is computer simulations of DNA condensation. So over to you, Prof. Serdal. Thank
you. Thank you very much for this kind introduction, and thank you for the invitation. I wish I would come in person and have a chance to interact with you guys. And also, I would like to see you here in Abu Dhabi. So the topic of my talk is computer simulations and using computer simulations to understand a phenomena that is crucial for life, which is DNA condensation. And before I start my talk, I'd like to give a brief overview of what I'm going to talk about. So the DNA condensation cannot be understood without understanding the counter ions and their distributions around the DNA double helices. I'm going to spend a lot of time to give a perspective on how we understand the ions around the double helices of DNA and RNA systems. And then I will talk about the DNA condensation mechanism and how we use molecular dynamics simulations to basically probe these interactions and come up with some lessons that we can develop better physical models to explain the phenomena. So as you may all know, that DNA and RNA are highly charged polymers. And these highly charged polymer structures, when we look at the X-ray crystallography structures of the RNA molecules and DNA molecules in the protein data bank, we oftentimes see there are ions attached to certain positions. These ions most of the times are magnesium ions, but we also see potassium and sodium ions around the nucleic acids. And these ions, some of them are binding to certain places that are important for catalytic activity. Sometimes they can be found in positions that is not doesn't have high charge density. So we, based on this, we classify the ions based on how specifically they bind to certain positions. And specifically bound ions can be monitored by methods like NMR spectroscopy, X-ray crystallography. But there are other ions, majority of the ions that is surrounding the DNA and RNA that serves as non-specific roles in stabilizing these complexes are called diffusively bound ions. So oftentimes it's very hard to detect and monitor and probe these kind of ions, but we know for sure that they're present because of the theoretical models that we have basically suggest that the ions that we need to see is much more, it should be more than that we are seeing in the crystallography. So the second group of ions can be studied by a method called small angle X-ray scattering. And I'm gonna discuss like how this method is basically works. So in this method, we send X-ray to a solution of a nucleic acid system, which the solution, the X-ray scatters from the DNA and ions, and then creating a profile, as you can see on the right-hand side. So what we do is we scatter the X-ray in two different energies. One energy is above or below the absorption energy of the metal ion that we are interested in, and the other one is just on the same energy level. The one that is on the same energy level get absorbed by the ion, so the signal comes primarily from the DNA or RNA and the water around it. And the first one comes from the entire system. So if you take these two scattering profiles and then subtract them, and then you ended up with a signal that primarily comes from the ions of interest. So this way you can look at through the scattering, you can look at the distribution of ions around nucleic acids. This is the only method that can report us information about the counter ions that are important for the DNA condensation and RNA DNA stability. So from an theoretical point of view, the way that we study these ion distributions is through the electrostatic theory. And here I'm showing a DNA molecule that you can see on the left-hand side. It's comprised of phosphate backbone. The backbone is highly negatively charged. Each phosphate group here has about like -1 charge. And then this is surrounded by the two regions. One is called the minor groove and the major groove. So the simplest approach to model the DNA is a cylinder or a rod. And the rod has charges like -1 charges along the DNA, with the charge density is basically monitored by the parameter called L. Okay, so if I have a high charge density, my L becomes smaller, and if my DNA, for example, goes to a single strand, then the L grows. So this way, the charge density can change as the conformation of a molecule can change. So, so the first and the most important theory of, I would say, the counter ions around the nucleic acids is developed by Manning and Osowa. And so what they have figured out, like when they first set up this system, saying that, like, let's look at an ion and its interaction with a rod of a uniformly charged density. And so we can write the potential energy of this system as I wrote it here. And then and we can basically define a length scale, which is called the Beerman length. This is the length at which the intermolecular, the distance, the energy between the electrostatic forces become equal to the energy of the thermal forces. So this is basically, this shows that how electrostatic interactions is going to compare with the thermal fluctuations in the system. And for physiological conditions, 150 millimolar sodium with room temperature, the Beerman length is about 7.1, which means basically two sodium ions, when they are below 7.1, their electrostatic repulsion is above the thermal fluctuation, which is KT. So we can, now we know the potential energy for one ion. What we can do is we can write the partition function of the system. And when they write the partition function of one ion around the rod of a uniform charge density, they discovered that this partition function diverges. Like if you take this and then I give it as an exercise to my students. So if you try to integrate this, it does not go to a finite number, which is a problem. So what because of the divergence of the problem, that they come up with this conclusion saying that suggests that the sum of the ions needs to condense on the surface of the DNA and then renormalize the charge. And the renormalization should be such that the net charge density becomes equal to the barium length. And this idea, this simplistic idea, come up with an interesting and simple result, which basically says that for a double helix, for example, where L, which is the charge density, is around 3.2 angstrom, the effective charge on the phosphate backbone has to be 0.25, which means 75% of the charge on the DNA is screened by this, by the counter ions that are serving to renormalize the charge, to make the partition function to be finite. So this idea can be basically like the simplistic argument can be further elaborated by a theory called Poisson-Boltzmann equation or Poisson-Boltzmann theory. So this, according to this theory, basically like we can take our rod again, and the rod, let's say that our rod has a net charge of rho. So according to the Gauss theorem, the electrostatic electrical flux can be given by this equation. And then because electrostatic field, electrical field can be written as the gradient of the electrostatic potential, I can write this equation, which is called the Poisson equation. So, and I also know that like if I'm R away from my rod, that the distribution of charges needs to be renormalized or weighted according to the Boltzmann distribution. And Boltzmann distribution is basically says that, so the weight, the probability of observing an ion at the position R is equal to the probability of observing an ion in the bulk times the energy weight that comes from the electrostatic potential at the point divided by the thermal energy. So what we can do is now, like we have two equations. One says that at a particular distance, the distribution of ions should be according to the Boltzmann law. And then the other one says that the electrostatic potential is given by the Poisson equation. So we can combine these two. And the combination of these two allows us to basically solve the electrostatic potential based on the solve the differential equation. And this differential equation is called the Poisson-Boltzmann equation. And Poisson-Boltzmann equation that I wrote here is basically a general geometric form. So I can write it in a cylindrical coordinate. And then if the charge density is low, or I'm only studying monovalent or divalent ions, I can linearize this differential equation and then end up with this one, which I can easily solve. And then here is what I get as an electrostatic potential along as a function of distance from the surface of the rod, which represents the DNA. So this equation, I can plot this equation, and you can see if I use a linear Poisson-Boltzmann versus Poisson-Boltzmann equation, then this is how the electrostatic potential decays as a function of distance from the surface of the DNA. So this is a theoretical. This is the theoretical approach to treat the counter ions and finding their distributions around the nucleic acids. And there is another approach which is more exact and more detailed, which is molecular dynamics simulation. In molecular dynamics simulation, what we do is we represent the interactions by using an empirical potential. Here, I'm writing an empirical potential of a molecule. Okay, so I have terms like the bond stretching and bond bending, like these are harmonic potentials. Dihedral angles can be represented by Fourier sum of angles. I have non-bonded interactions between atoms that are not covalently bonded, which is treated by Lennard-Jones, and then I have Coulomb term. So I can, by using this functional form and all the parameters trained by an independent resource, I can model a DNA. And then I can also model the ions, and I can also model water, and then I can put them all together in a simulation box. And then what I can do is, once I know the potential energy, then I can find the gradient of potential energy, which is force. And then once I know the force, once I know the initial position and the velocities, and then I can solve the Newton's equations of motion and iterate this procedure millions of times. This is called the molecular dynamics simulation trajectory. When I do that, I can see my DNA molecules like making conformational changes. My ions and my water molecules are going to basically move. And then if I run my simulation long enough, then I can collect my trajectories and then basically calculate any equilibrium distributions, any equilibrium properties that I'm interested in. In this case, I can calculate the ion distributions. And then from my trajectories, I can also compute the scattering amplitude. Then I can go back and then compare with anomalous small angle X-ray scattering. So this way, we can have a precise, direct way of comparing experiments with the theory. So here is what we did and our comparisons with this anomalous small angle X-ray scattering that was measured by Louis Pollack lab from Cornell. You can see that for monovalent and divalent ions, we can get quite good agreement from the molecular dynamics simulation. So,
sorry, Serdal, yes. Can I ask you a question, please?
Please go ahead.
Could you go back to the previous slide?
Yes.
So what is the f of q exactly from the simulation? Is it the g of r? Before you transform the g of r.
F of q is the form factor, which is the electron density. But the electron density slightly changes as the wavelength changes. So it's
okay.
So that factor is within the Q range that we are. It's only changing like 2%.
Okay.
But this is basically the electrons. Yeah.
I see. So I missed. So what exactly are you getting computing from the simulations that allows you to get this small angle scattering spectra?
So the simulations are giving me atom
positions.
Atom positions. From atom positions, we basically obtain the pair distribution function.
Okay.
And the scattering amplitude basically is the Fourier transform of the pair distribution function. So that automatically gives us, like, from the real space, we can go to the Fourier space, which is projected in the scattering curves that I have here. And then what we can do now, then once we have a good agreement, then we can say that, okay, once we agree, then we can convolute the small angle X-ray scattering and then go to the real space again, which is, then we can look at the ion distributions. So this way we can see how the I of Q reflected profiles are in real space look like. And you can see from here that, for example, if you have a rubidium, which is a monovalent, the ion distributions are forming a bigger cloud when you compare it with a divalent system. In a divalent case, the counter ions are basically like localized in the major grooves. In the case of the monovalent ions, they are basically forming a diffusive cloud. And this, and we can also, from the simulations and also from experiments in a different way, we can compute how much of the charge is screened through the counter ion condensation. And then we can compare these values with basically the mean field theories like the Poisson-Boltzmann, and also with the counter ion condensation theory.
So, Serdal,
yes.
Quick question. So for sodium and potassium, you expect it to behave like rubidium. And for calcium and magnesium, do you expect it to behave like strontium?
Yes, with
Okay.
Yeah, I mean, it will
Because in our
behave like
bodies
this.
we have, yeah, in our bodies we have sodium, calcium, potassium, magnesium, not rubidium, strontium. Right? Okay.
Exactly. Rubidium strontium is a necessity for the anomalous small angle X-ray scattering, because the X-ray, there is the wavelength are not the wavelength of the magnesium and calcium is not accessible through this method. So that's the reason why we are basically bound with heavy atoms. But that's a limitation of the experimental technique. But through simulations, we cannot study anything.
Okay. Yeah, but for this, so for sodium and potassium, they will behave probably like rubidium, right? Because they're monovalent.
Yes.
Okay. And then calcium and magnesium like strontium, right?
Yeah, I mean, we see slight differences when we look at the cation size effect. The valence is the most important factor in determining the ion distributions.
Okay.
But the cation size also has some minor impact, which we can see through simulations, but there is no experiment that directly probe the differences between the ion sizes.
Okay. Thank you.
You're welcome. So what we can do is once we see the ion class here, we can basically characterize ion class in a different way by using concentration profiles computed in the cylindrical coordinates. We basically create a cylindrical shell and then count how many ions we have at that certain region, and then we bin it. And once we do that, when we can see that the ions basically bind to two different regions, and one of them is called the major group, and the other one is called the minor group. And the affinity of ions to different regions depends on the valence, depends on the valence, and we can also see the decay length is different depending on the valence. So these kind of things are what the molecular dynamics simulation coupled with an experiment provides. So we can also go back and then say, how accurate is the Poisson-Boltzmann theory to describe the DNA and ion interactions? And this is basically the comparison of different Poisson-Boltzmann techniques with different parameters compared with the monovalent ions. You can see that in the case of monovalent ions, the Poisson-Boltzmann theory is pretty much okay. The decay is perfectly accurate, and then the peak positions it's getting the peak positions correct, almost correct. But in the case of magnesium, the divalent ions, we started to see deviations where the mean field theory starts to deviate from molecular dynamics simulations.
Serdal, just a clarification. So this is standard Poisson-Boltzmann, not linearized, right?
This is, I don't have the labels here, but the blue is linearist.
Okay.
And then the green is non-linearist. And then the red is size non-linearist with size effect included.
Okay, so that's let's say the highest quality mean.
The highest quality, exactly.
Okay, so clearly the linearized is of course very bad.
Yes.
Yeah. Okay. Cool. Thanks.
So electrostatic theories that we have. So the question now is is the electrostatic theories that we have is enough to study biological processes like DNA condensation? And what's DNA condensation is basically, it's a very complex process and it's a very hot topic nowadays. So this is the DNA of about 2 meters long. basically goes a multitude of processes and leading to a very complex structures called chromosomes. And during this process, it first formed the nucleosome core particles, and then the nucleosome core particles form the chromatin fibers. Fibers form the loops, and the loops basically fold one more time, which and then form the chromosomes. And some of these processes are governed by molecular motors like the histones, but the majority of the process is governed spontaneously. And there is how this complex condensation mechanism is controlled and orchestrated spontaneously is still an unknown question. So what we would like to study is like you're trying to find out some fundamental principles governing this process, and to further make this the understanding of the biological process to be possible. So here is another example where the DNA condensation is really interesting and happening all the time. Is a virus. So if you look at the head of a virus, you see that the DNA, and this DNA needs to be compressed in an environment in a cavity of 70 nanometers in size, and the DNA itself is around 140 times 10 to the 3 nanometers. So it's like huge compaction needs to be done, and the compaction needs to be done in a proper way so that at the end of the day, when the virus needs the DNA, it can basically take the DNA out without any problems. So the way that is happening is basically shown in this Crow-EM structure, which was published in 2016, is that the DNA inside the virus is forming a super compact, but at the same time ordered structure. So you can, from here, you can basically literally see the DNA, how the DNA molecules are packed inside the virus. And if you look at carefully to the layers, then we see that the DNA inside the cavity is forming an hexagonal pack structure. So the question is like, what drives the DNA molecules, which are highly negatively charged, even with the presence of the counterions around them, to form such compact structures and the hexagonal structures. So people have questioned this for a long time, and one methodology that was developed to study the DNA interactions in different conditions to mimic the confinement effect is done by Parseghi and Rao from NIH. So what they do is they take the DNA arrays and then they put it in a PEG solution. So the DNA inside the PEG solution is put in a membrane, and one side of the membrane there is water ions and the DNA molecules in the presence of PEG. In the other side there is only water and ion, and the membrane is permeable to the water and ion. So this setup ended up with a difference in the osmotic pressure, and I'm going to explain why the osmotic pressure difference is happening in the next slide. So this osmotic pressure difference is a way that they can directly measure the intermolecular forces between DNA molecules. So the reason why there is a change in the osmotic pressure is basically the following, called the discovered by the Asakura and Osawa, is called the depletion forces. So imagine that the two DNAs are far apart from each other. They have an excluded volume, which is occupied by the PEG molecules. When the two molecules come closer to each other, they create a region which is inaccessible by the PEG molecules. And the PEG molecules used to reside in between the two interfaces, leave the surface and then go inside the solution. And when it happens, the accessible volume to the PEG molecules increase. The increase in the solvent accessible area creates a higher translational entropy for the PEG molecules that creates the partition function or the Helmholtz free energy to go lower values, and the change and Helmholtz free energy, derivative of the Helmholtz free energy, gives us the osmotic pressure. So we see a change in the osmotic pressure because of the increase in the translational entropy due to the PEG molecules that are no longer belong to the excluded volume. So using this technique, what they do is they measure the osmotic pressure, which reports how many of the PEG molecules are basically exposed to the solvent from the depletion zone. They also measure at the same time the distances between the two DNA pieces through the X-ray diffraction, and then by plotting the pressure as a function of interspacings between the two DNAs, they observe an interesting behavior can be represented by like a van der Waals fluid, like a van der Waals equation. So at certain conditions, the DNA molecules act like repelling each other, which is the expectation that we have. And at certain salt conditions or sequences, then people start to see a two-phase behavior, where the DNA goes from one packing condition to another packing condition. So using this osmotic stress method, people have studied different scenarios. Like, for example, this is when the genomic DNA is present in the presence of sodium. So you see that the dots here representing different conditions of the different salt conditions, but all depends on the sodium. And what we see is that in the presence of sodium, DNA always behave like a repulsive. And the CC and PB here represents the counter and condensation theory estimate versus the Poisson-Boltzmann theory estimate. So you can see that both of them are slightly off, but they get the general trend accurately. So if we go to the divalent ions, on the other hand, so we see that both theories also not giving rise to a good agreement, but at the same time, PB is slightly better. And the osmotic stress measurement basically suggests that there is a repulsion even for the genomic DNA in the presence of magnesium also. So the monovalent ions and divalent ions are not sufficient to condense DNA. The DNA molecules repel each other according to these measurements. But when we have polyvalent ions in solution, so for example, the spermine, spermidine, cobalt hexamine, we start to see the attraction happening. So this is the place where the physiological condition where is also like in our physiological in the physiological condition, we have a lot of amines like spermine, spermidine present in the nucleus that basically suggest that they are helping the DNA molecules to condense spontaneously.
Yeah, but Serdal, with spermine and spermidine is not only child-child interactions, right?
It is, yeah. They have different mechanism to basically bring the DNAs together, and so what people like, because of their shape, they're like a rod-like, and they can go inside the minor grooves and then bridge the two in a non-electrostatic mechanism, right?
Okay.
Yeah. So, but what people also have observed is that, like, unlike magnesium, a bigger size ion, a transition metal ion, manganese, can also lead to condensation. And also, recent studies also show that while the genomic DNA is repulsive, also the poly-A, which means like one strand is completely A, the other strand is completely T, can also be attractive. So basically, like there are a lot of other scenarios, which I'm just skipping for the sake of time, but we see a lot of different ways that the DNA can go to condensation. And the mechanism, like the theoretical foundations of why this is happening is not clear yet. There are many theories proposed to explain this, and I'm just going to list them, like the Osopha-Contra condensation theory, Parsekian-van der Waals interaction theory, the zipper model, and the Wigner-Crystal model, and the ion bridging model are the ones people oftentimes use to explain one experimental condition, but these methodologies most of the times fail when you study your system in a slightly different condition. So they don't have pretty much predictive power in explaining the DNA condensation phenomena. So like to give, like this is going to be important for the closest thing that we have found from the repertoire of the ion condensation theories is the zipper model. So the zipper model basically suggests that the counter ions that is condensing on the DNA, they're basically condensing specifically on certain positions and creating a charge patterns on the DNA surface. And these charge patterns, like they're binding to the major groove, and then they're leaving the phosphate backbone naked, according to this theory. So once that happens, like so the free energy on top of the cylinder mean field Boson-Boltzmann term that I described before, we ended up another term which they call the free energy of the helical interaction, which is happening because of the counter ions that is specifically binding to certain regions. And then these ions are also dynamic, so they can basically they can explore different positions. So this is basically the accounts for the free energy of the translational motion along the DNA backbone. And by using this theory, they come up with a conclusion saying that the attraction can be possible in certain conditions when the occupancy on the major groove is above a threshold value, and the hallmark of the attraction is to see an azimuthal order. Like the angles that the two DNAs should align themselves in such a way that they should have an angle correlations on the DNA arrays. And the second most recent theory is basically says that especially for the ions for the polyvalent systems where spermine and spermidine, the mechanism of DNA condensation is through ion bridging rather than rather than these ions are forming charge patterns on the surface, and they have reported that there is no angle correlation, which suggests that the zipper mechanism doesn't hold for the polyvalent ions. So, so we would like to come up with a general mechanism and to understand the DNA condensation mostly for point size systems. And as I showed before, like none of these theories can explain why the DNA condensation is sequence dependent, why some certain sequences are showing attraction while others are showing repulsion. So as a tool, just like before, we're relying on molecular dynamic simulation. In molecular dynamic simulation, what we do is We are taking it to DNA pairs, we put it in solution, which we mimic exact this salt conditions as the experiments. And then we monitor the angle as a metal angle, and then the DNA interhelical distance. So what we can do is like we can calculate the free energy along these two degrees of freedom to enhance the sampling and to make sure that we reach thermodynamic convergence. What we do is we use a technique called metadynamics. In metadynamics, what you do is you add a biasing term to the Hamiltonian, and this biasing term is basically a probe that monitors the convergence and at the same time allows an enhanced sampling. So every time this biasing term is written here, it's basically a Gaussian. Anytime we visit a particular value of d and theta, we are adding a Gaussian on that position. And that makes the energy, the potential energy slightly higher. And then every time, and then this way, as we run our simulations long enough, the free energy, we construct the free energy by just looking at all the biases we add to the system. So here's like a depiction of how this is happening. The advantage of this technique is it allows us to overcome the barriers and then explore conformational space more efficiently. And at the end of the day, we get the free energies, as I said before. So here's a simulation of DNA in magnesium chloride. And then I'm going to show the DNA simulation in spermidine. So once we run the simulations and the systems converge, we can look at we can look at the free energies in one dimension. I write it here. So the DNA, this is the genomic DNA in the presence of magnesium, and this is my free energy as a function of interspacings between the two DNA. Here I'm plotting it as a polar plot, which I show here the angle and also distance between the in one figure. You can see that the blue regions represent the lost energy. As you can see, for a mixed DNA or the genomic DNA, the distances are further away for the blue region, which means that the two DNAs are repulsive with each other, and there is no angle correlation, so they can probably obtain almost any angle possible, except there's a forbidden zone. We don't know why we have this. If you look at the spermidine, on the other hand, we see a deep minima, and this deep minima also reflected in the polar plots, as the system not only goes to the condensed phase, but also prefers angle, like two specific angles, which I represented here. So there is angle correlation in also the condensation happens in the presence of spermine. And also, if you look at the sequence effects, we see that the mixed DNA, as I said, shows the genomic DNA in the presence of magnesium shows repulsion. If I take a poly-AT sequence, it shows attraction, just like the experiments report. And we can also see the angle correlations. Angle correlations are key because it basically suggests that the system is like a zipper-like model. And if you compare this with the experimental findings, we were able to show that this is correct, and then we were able to show that this is we can get agreement with the experimental results of this one, and then this one. I'm not going to show, but we using simulations, we were able to capture this and this one. So now we can capture experimental results accurately with every salt condition and sequence using molecular dynamic simulation. So the question is like, what is the mechanism then? So to understand the mechanism, we look at certain, we probe certain parameters. Like the first one we probe is the water, because some studies suggest that it's the hydrophobic effect that's leading the DNA molecules to come and condense. So when you look at the water, what basically happens when the system far away from each other, it has a solvation shell. The solvation shell is included ions and water molecules. So the system goes from a unbound state to the bound state. What happens is that the number of water molecules goes down. So basically the water that is surrounding the DNAs are going to the solvent. When they are on the surface of the DNA, they used to have less degrees of freedom, become when they go to the solvent, their translational entropy increases. So for water, if I have a larger solvation shell or a small solvation shell, we both observe this kind of behavior. And the mixed DNA and ATDNA basically show differences. So their hydration levels are very different from each other. But the general trend remain the same, which means like as they come closer to each other, they release water. The release of water basically enhances the condensation, helps the condensation to happen because of the translation, the gain in the translational entropy. So we also monitor the magnesium ions. Magnesium ions, this is the outer shell of the magnesium ions, slightly reduces, and there is no difference. There is not much difference between the ATDNA and mixed DNA to basically to make them different from each other. And but the second layer, which is this is the distance, like the shorter distances, what really happens is that the magnesium ion population increases. So what really happens is that the magnesiums that is in the outer shell starts to migrate to the inner shell as the DNA comes closer and closer to each other. So this migration can give us a clue, but by just looking at the numbers, we don't see any major difference between ATDNA and mixed DNA. We can see that like this is the reason. So to understand the difference, we also look at the radial distribution function. This is basically a function that allows us to probe the molecules in the neighborhood. So the peak suggests that the peak position suggests the first salvation shell. The second peak position suggests the second salvation shell. So if we look at the DNA and separate it into three different regions, like the phosphate backbone and minor groove and major groove, we see that there are differences in the binding modes. It's not the number that is that's significantly different, but the way that ATDNA binds to the major groove or the phosphate gives it a stronger stability. And the reason for the ATDNA shows such a unique behavior can be seen on the left-hand side, like the ion distributions plotted in mixed DNA and ATDNA. What we see is that in the case of mixed DNA, we have two layers. The first layer is this layer is the major groove binding, and the second layer is around the phosphate binding. And this binding, these two modes of binding is continuous, and it is very strong and localized around the helium. On the other hand, in the case of mixed DNA, the major groove binding is very weak. You can see like a few spots here and there. The majority of the binding happens only on the phosphate backbone. And as a result, not because of the total number of ions are different, but the way that the ions are distributed and localized around the DNA, it creates an electrostatic interaction of mixed DNA, ATDNA to be a lot lower than the ATDNA. And also, a second kind of stabilization that we see is that that happens because of the translational entropy. So what we did was we also calculate the translational entropy change when the system goes from the unbound state to the bound state. And what we observe is that the DNA conformational entropy does not change, but the ions translational entropy show differences. And these differences also sequence dependent. ATDNA shows very little translational entropy penalty when the DNAs come closer to each other. The ions remain very mobile and then can go between the two DNAs. But in the case of mixed DNA, they are trapped, and this localization of the ions on the phosphate backbone leads to an increase in the entropy, decrease of the entropy that basically penalizes this stability of the system. So we said that, okay, why the poly A is showing attraction? Is it because that the adenine or thymine has some kind of unique chemical interactions or specific interactions with the cations? So we studied two systems, one with exactly the same chemical composition. One is the poly AT, the other one is poly A and poly T. So here is the difference between the two. And then we measure the osmotic pressure, and then we compare with the experiments to see if we are doing right. So you can see the dots representing the simulation, and the lines are representing the experiments. We see a very good agreement between the two approaches, but we also see that, interestingly, that the poly A, poly T is attractive and poly AT is repulsive. And the reason, again, what we found is that it is the distribution of ions on the surface is making the difference between the two. So in the case of AATT, the two negative groups that is on the surface, which one is called N6 and N7, are creating another electronegative layer that accumulates the ions and then leading to an effective like two-layer systems that leads to stability. In the case of the ATDNA, ATTA, the ions, the two ion, even the two binding sites, even though they are present, they're smeared out on the surface of the DNA, not creating enough charge density for the ions to come and bind. That leads to the disruption of the second layer that I described, and that leads to the instability of the complex. So based on all this analysis, we come up with a simple model that allows us to basically explain the DNA condensation. And according to this model, this simple model has two components, and one of them is the mean field component that includes the cylinder interaction. So we use a Poisson-Boltzmann analytical theory to represent the interactions between the two rods. Each rod is interacting other one with an effective potential, with an effective charge. The effective charge is the charge that is after the Manning's condensation happens. So, for example, the effective charge of a DNA is 0.25 per base pair. And this is the repulsive term in our system. And because of the helical nature of the rod, what we also in our model we also include the ion correlation. So the ion correlation term is the following. So when an ion binds to the phosphate group, it makes the phosphate positively charged. When it doesn't bind, it creates a hole, and the hole is negatively charged. And the ions basically hop between the two binding sites, and then stay sometime and in the bound state, sometime in the unbound state. So these two, and then once they are like opposite to each other, they create an attraction. And then what we do is, like in our model, we have a term called the correlation term. The correlation term, we count all different possibilities of the hole and the occupied site formation. And then we use the grand canonical partition function, and then we calculate the total partition function, and then take the derivative of partition function and calculate the forces between the two. And this allows us basically to see the two regime, I would say. The regime, in the first regime, that we see an attraction. Attraction happens when the parameter F that I have here, which is the fraction of the phosphate backbone occupied by a magnesium ion or polyvalent ion. When F exceeds the threshold value, which is around like 60%, then we see an attraction. If it is below that value, then the ion fluctuations does not lead to attraction. So this theory that couples the Poisson-Boltzmann and the counter-ion condensation theory with the ion fluctuations that properties come from molecular dynamic simulation allowed us to explain almost all of the cases that we have seen so far in the DNA condensation. So I'm going to stop here because I'm already late. I'm going to skip this part. So my conclusions are basically that the counterions are the essential and important part of the nucleic acid assembly and structure. And the computer simulations are one of the best ways of looking at the ions and their interactions in a unified framework. And by looking at the molecular dynamic simulation and the ion counter-ion condensation, we can explain the DNA condensation, its sequence specificity, and how it interacts with its binding partners. We see that the specificity comes because of interactions. Sequence specificity comes because of the interactions that is on the surface dictated by the different sequences. And what makes a DNA molecule to attract another DNA molecule is the dynamic coupling between the cation motions that is at the interface of the two. And these rules that we observe can be used basically to create DNA assemblies and to understand a lot of processes happening, like the DNA positioning and the recognition of DNA molecules by the binding partners and the DNA packing. With that, I'd like to thank my group members. The work that I present today was done by Wei Wei, my PhD student. He's a brilliant PhD student. And I'd like to thank my other group members for the discussions. Some of the work was done in collaboration with Pollack Lab from Cornell. And then the osmotic pressure measure simulations is in a collaboration with Professor Chui from George Washington. And thank you for your attention.
Thank you very much, Serdal. Thank you for the interesting talk. So now we'll take questions. So if you have questions, you can unmute yourself and ask, or you can put your questions in the chat. So there's a question already in the chat by Moses Wambua. Do you see the question, Serdal?
I can't see, but let me see.
Oh,
How can
no,
I
you
make
want to. Okay, let me post the question and send it to you.
Maybe if I stop share, I can see the question, or it's
No, I think
oh,
you,
okay.
sent
Now
it.
I can see. Now I can see the question. Okay.
Yeah, I sent it.
So the first question is, I missed the first few minutes of the presentation. Can these charge interactions influence DNA polymorphism in a population of organisms? If yes, I see great potential in population genetics. I mean, this is kind of like a interesting question. So what the modulation of interactions and the DNA packing very much depends on the environment and soil conditions, and many of the different organisms have different salt conditions. Like some organisms have very high magnesium concentration. Even like within the cell, the nucleosomes, for example, has a high magnesium concentration and potassium concentration, versus in cytoplasm is very little magnesium concentration and sodium concentration. So each species, I think, has a unique way of modulating these interactions through the different compositions of the ion distributions that they possess. And I think this, because we don't see the impact of the ions in experiments, we don't know like their direct role in many of the biological processes, and there is a lot more to learn by doing like comparative studies between mechanisms between the organisms.
Okay, thank you very much, Serdal. Professor Ali Hassan Ali has a question.
Thank you, Omo. So thanks a lot very much for your talk. I had a more practical question about doing molecular dynamic simulations with fixed point charges. So I know there have been discussions in the literature about that basically most classical potentials, especially for the ions, they don't include electronic polarization, of course.
Yes.
And so people often correct for this by scaling the charges. And I mean, looking at your structure factors for rubidium and strontium, it looks like one could get away without scaling the charges. So I was just wondering what your thoughts are on this, and in general, what is the importance or not perhaps of electronic polarization in these systems?
I mean, I think from a physics point of view, we need the polarization to get the exact physics behind it. But from a practical point of view, not because of the computational cost, but because of the poor parameterization that we have right now. So polarized force fields are doing much worse than the point charge force fields. And that is basically the bottleneck, I would say, in the reason why we are not using the polarized force fields in my group. Okay. And we have tried different cases and we never get experimental agreement. So there is this is because that non-polarized force fields have been used a lot and optimized a lot. Mm-hmm. And there is only a few groups working on polarized force fields, and their efforts is not enough to basically come up with a set of parameters that can represent the biomolecules. So I see. From that point of view, like maybe in 10 years or so, we should be doing all the simulations again, and then look at what we have missed before when we were like,
okay.
But at the moment, like even this seems to be reasonable enough. So we try to basically like base all our conclusions on experimental verification. So if we don't get an experimentally correct observation, we don't believe that the results that we get.
Yeah, okay.
Yeah.
Can I ask another question, Omar?
Please go ahead. Yeah.
Yeah, sure.
Okay. So it's another, well, maybe an annoying question about the free energy calculations. So when you do the metadynamics in these types of complex systems, it looks like you're getting away quite well with using a collective variable, a single collective variable, right? Or and you know, let's say these things are very high dimensional objects. So, but it looks like, so for this problem, you have the slowest degree of freedom is basically captured very well by the distance between the DNA molecules or something, right? So, is there, from the experimental side, do you have some validation that allows you to say, okay, you are okay with using that collective variable?
Yes. Yeah, I mean, that's a good question. So let me go like and show. So here, so what we do is like, the reason why we choose interspacings and the angle is basically, we think we would like, not because they were slow, but this is like representing the reaction coordinate, we believe. Like the two DNA molecules that is infinitely long, and in a packed environment, they can only do distance changes and then rotate, right? I mean, like they can rotate like this. This is identical to just rotating one of them. And these are the two things which we would like to project the free energy surface and then look how the projection looks like.
I see.
And then, yeah, so that was from a physics perspective rather than like thinking that they're slow, but we find out that if we have these two degrees of freedom, we can project the free energy landscape. So once we get the free energy coverage, then we compute from the two-dimensional free energy landscape, we reduce it to one-dimensional free energy landscape, which is which I can show you here. So this kind of free energy is basically like, it's a reduction of the two-dimensional energy landscape to one-dimensional energy landscape.
I see.
And then once we get the one-dimensional energy landscape, then we calculate the osmotic pressure. Osmotic pressure is basically the derivative of the energy with respect to the distance. And then we go back, and then this is basically, it gives us the average force. Okay. And then if I scale the average force with the hexagonal geometry, I can get the osmotic pressure directly from the simulation.
So I mean, just in this regard, you don't have problems with the kind of elastic bending of the DNA.
Yes. So what we do is we are mimicking an infinitely long DNA.
Okay.
To mimic an infinitely long DNA, what we are doing is we just I'll show you. Sorry. Yeah. So
got it. It's periodic. Okay.
Exactly. If you set up your simulation box that has only two terms, then it goes infinity.
Makes sense.
And
so
this way you don't have the bending degrees of freedom. And then once from the free energy, you get the average force. And then from the average force, you get osmotic pressure. And from osmotic pressure, you compare with the experiment. And I mean, when we compared with the experiment, we were shocked because the agreement was quite good.
Yeah, right. Yeah, sure. Yeah. Okay, no. Okay, now I understand what. Okay, thanks. Thank you.
Okay.
Thank you very much, Serdal. Any other
Thank you.
questions?
Any other questions? Yeah.
You can drop your question in the chat, or you can unmute yourself and ask. Okay. Okay, Serdal, I see no other question. Nobody's hand is up. Okay. So yeah, so thank you very much again, Serdal, for the very nice talk. So the video will be available online, YouTube. If you send us your slides also, we have a place where we can put it for other people to be able to take a look at later on. Yeah. So let's thank Professor Serdal once again. And then thank you very much, Serdal.
Thank you. Thank you for having me. It was a pleasure for me.
Yeah. We are very happy to have you also. Thank you. Yep.
Thank you.
Yep. Okay.
Bye.
Yep. Bye. Yeah.